Hello students, welcome to EPG Pathshala. I am Dharmila from Jamia Hamdard, New Delhi. In this module, we are going to discuss the pharmaceutical validation part 2. Paper Pharmaceutical Validation part 2. As in previous pharmaceutical validation part 1, we discussed about the validation and importance of validation, documentation associated with validation and approaches for validation. And now in this module we will learn about the classification of validation and its importance, various types of qualification, validation of facilities and processes and their benefits. Since the validation activity covers a wide variety of processes, equipment, utilities and methods, etc., validation can be classified as follows: qualification, validation of facility and equipment, calibration of equipment, validation of process, validation of analytical methods, validation of utilities, validation of cleaning procedures, validation of computer systems. Qualification or validation of facility and equipment. Qualification and validation are essentially similar concept. Qualification is considered to be the act of planning, carrying out and recording the result of tests on equipment to demonstrate that it will perform as intended. Qualification should be completed before process validation is performed. Commissioning and qualification is the foundation stone on which the process validation is built. It is function of facility, equipment or utility which determines what level of commissioning and qualification is required. Basically, these can be classified as: direct impact system is expected to have an impact on product quality, indirect impact system is not expected to have an impact on product quality, and no impact system gives no impact on product quality either directly or indirectly. In case of indirect impact system and no impact system, the organization needs to follow good engineering practice. However, in case of direct impact system, supplementary qualification practices need to be followed to meet the additional regulatory requirements of authorities. The different stages in the performance of qualification include design qualification, installation qualification, operational qualification, performance qualification and requalification. Design qualification. Design qualification constitutes the assurance that the premises, supporting utilities, equipment and processes have been designed in accordance with the requirements of GMP. This will be the first step in the qualification activity. Next is installation qualification. Installation qualification is an activity regulated by regulatory authorities and considered as a pre-requirement before operational qualification. Installation qualification is the documented verification that all the aspects of a facility, utility or equipment that can affect the product quality adhere to pre-approved specifications. The purpose of installation qualification is to establish that the critical components are installed correctly and is in accordance with the design documentation. There should be a protocol which describes some specific attributes of the facility or equipment through which one can demonstrate that the installation is done as per the design document. The basic requirement for writing an installation qualification protocol consists the following: validation master plan, user requirement specification, suppliers drawing and specification. The content of an IQ protocol are generally as follows: approval page for document review and approval, prerequisites, activities to be completed prior to the execution of the protocol, objectives mentioning the purpose of the IQ, system description, equipment number, location, system functionality, etc. Responsibilities describe who will perform what and when. Engineering documents, list of documents required to support the system such as utility verification, control system verification, etc. Acceptance criteria, deviation and corrective actions, how to address the deviation if any occurred and the personnel responsible for that. Summary of IQ, references and appendices. IQ protocol should be numbered, dated and approved for issue by appropriately authorized personnel. During the IQ process, it should be verified that the item matches the purchase specification and that all the drawings, manuals, spare part list, vendor address and contract numbers and other important documentation are available. Overall process can be streamlined by giving proper training to end users. This training program should cover the purpose of equipment, applicable SOPs, etc. All training needs to be documented. Upon execution of installation qualification as per pre-approved protocol, the report should be generated and key findings should be summarized. The report should be reviewed and approved before the operational qualification commences. Operational qualification. Operational qualification is associated with the performance of the equipment to ensure that the function of machines, measuring devices, utilities and manufacturing areas operates according to its operational specification in the selected environment. This ensures that facility, utility or equipment operates as intended throughout all anticipated ranges. Execution of operational qualification involves testing of parameters of facility, utility or equipment that determines the product quality. As in case of installation qualification, there should be a protocol for operational qualification. The basic requirements for writing an OQ protocol consist of following: equipment operation manual, functional requirement specification, supplier drawings and specification, standard operating procedures. The OQ protocol should be numbered, dated and formally approved. The content of an OQ protocol are generally as follows: approval page for document review and approval, prerequisites, activities to be completed prior to the execution of protocol, objectives, purpose of OQ, system description, description of equipment, its operation and any limiting parameter of the equipment, responsibilities which describe who will perform what and when, acceptance criteria, qualification test, equipment or instrument list, overall testing plan, SOPs, cleaning procedures, deviation and corrective actions, how to address the deviations if any occurred and the personnel responsible for that, summary of OQ, references, appendices. The OQ data should be formally reviewed and approved before the process validation can commence. Performance qualification. Performance qualification is done after both IQ and OQ have been completed, reviewed and approved. This is the final qualification activity before the process validation begins. Performance qualification integrates procedure, personnel, utility or equipment to verify that the system as a whole will produce the required output such as sterilization condition, contact utility, for example water for injection, clean compressed air, or environment, for example HVAC. As in case of IQ and OQ, there should be a protocol for performance qualification. The basic requirements for writing a PQ protocol consist of following: piping and instrumentation diagram, user requirement documents, acceptance criteria, standard operating procedures. The PQ protocol should be numbered, dated and formally approved. The contents of PQ protocol are generally as follows: approval page for document review and approval, prerequisites, activities to be completed prior to the execution of protocol, objectives, purpose of the PQ, system description, description of equipment, its operation and any limiting parameter of the equipment, responsibilities describing who will perform what and when, acceptance criteria, test equipment or instrument list, overall testing plan, SOPs, cleaning procedures, deviation and corrective action, how to address the deviation if any occurred and the personnel responsible for that, summary of OQ, references, appendices and analytical test results. The OQ data should be formally reviewed and approved before process validation can commence. Requalification. Once equipment is qualified, you have to verify the qualification at periodic intervals or after making certain changes in facility or equipment. Based on this concept, requalification is classified into two, that is periodic requalification and requalification after change. These changes include equipment maintenance, movement and repairs, etc. Calibration of equipments. Calibration is defined as the performance of tests and retests to ensure that measuring equipment, for example for temperature, weight, pH, etc. Used in manufacturing process or analytical procedure in production or quality control gives measurements that are correct within established limits. Equipment is to be calibrated according to written procedures to assure proper performance. Written records of those calibrated checks and inspections shall be maintained. These written procedures should include limits for accuracy and precision. Precision has no unit to measure and only indicates a relative degree of repeatability, that is, how closely the values within a series of replicate measurements agree with each other. Bias is the measure of how closely the mean value in a series of replicate measurements approaches the true value. The mean value is the number attained by dividing the sum of the individual values in a series by the total number of individual values. Accuracy. Accuracy is the measure of instrument's capability to approach a true and absolute value. Accuracy is a function of precision and bias. Generally, zero bias and high precision will lead to high accuracy. As per CGMP requirements, a calibrated program should be available. For each piece of equipment requiring calibration, the following information should be available: calibration standards and limits, responsibilities for performing calibration, intervals between calibrations, record keeping requirements and logs, actions to be taken when problem is identified. Preventive maintenance is also linked to calibration, as regular maintenance of equipment will help to keep the equipment in good working condition with calibration intervals. After calibration, the equipment should be properly labeled and should display the date of calibration and due date to perform recalibration. Personnel who provide the calibration and preventive maintenance should have appropriate training. Validation of process. Validation of manufacturing process is an important activity in pharmaceutical manufacturing which provides a high degree of assurance that the given manufacturing process will consistently produce products of predefined quality. Process validation is related to a life cycle concept. This links the product and the process development, qualification of the commercial manufacturing process and maintenance of the process in a state of control during the routine commercial production. Process validation should only be initiated after completion of qualification process. The extent of validation is linked to complexity of the process. Each critical process parameter needs to be defined during development and scale up stages, and validation protocol should challenge the critical parameters. All documentary evidences related to the process should be accumulated. Types of validation has been discussed in pharmaceutical validation part 1. Validation of analytical method. Before starting the manufacturing of any product, it is imperative that the specification and analytical test procedures of API, excipients and finished product should be available. The analytical procedure should be capable of giving accurate result in actual testing conditions, and this is ensured by validating the analytical methods. Critical factors that should be validated includes specificity, accuracy, precision, linearity, range, limit of detection and limit of quantitation, system suitability, robustness. Specificity. Specificity is the ability to assess the analyte in presence of other components, for example, degradants, placebo, etc., which may be expected to be present, such as impurities, degradation product and matrix component. Specificity has following implication for tests listed below: identification test to ensure the identity of the analyte, purity test ensure the performed analytical procedure allow an accurate statement of the content of impurity in the analyte, assay which provide an exact result which allows an accurate statement on the content or potency of the analyte in the sample. In case of assay, specificity can be proved by spiking pure substances with appropriate levels of impurities and or excipient and demonstrating that the assay result is unaffected by the presence of these materials, which can be done by comparison with the assay result obtained on unspiked samples. For the impurity test, the discrimination may be established by spiking drug substance or drug product with appropriate level of impurities and demonstrating the separation of these impurities individually and or from other components in the sample. If impurities or degradation product standards are unavailable, specificity can be demonstrated by comparing the results of sample containing the impurity or degradation products to a second well characterized procedure. These comparisons should include samples stored under relevant stress conditions, for example, light, humidity, temperature, acid base hydrolysis or oxidation. In case of assay, results should be compared. In case of methods for related substances, impurity profile should be compared. Accuracy. It is defined as the closeness of test results obtained by the analytical procedure to the true value. It is also considered as the difference between experimental value and the true value. This can be proved by applying the analytical procedure to an analyte of known purity or comparing the test results obtained with that obtained by a well characterized alternate method. Accuracy of the analytical method is to be proved across the working range of the process. Accuracy is calculated as the percentage of recovery by the assay of the known added amount of the analyte in the sample, or as the difference between the mean and the accepted true value together with the confidence interval. ICH recommends that accuracy should be assessed using a minimum of 9 determinations over a minimum of 3 concentration levels covering the specified range. Next is precision. Precision is an analytical procedure expresses the closeness of agreement between a series of measurements obtained from multiple sampling of the same homogeneous sample under prescribed conditions. The precision of an analytical procedure is usually expressed as standard deviation or relative standard deviation, that is coefficient of variation, of a series of measurements. Precision may be considered at following 3 levels. First is repeatability. Repeatability expresses the precision under same operating conditions over a short interval of time. Repeatability is also termed intra-assay precision. It should be assessed using a minimum of 9 determinations covering the specified range of the procedures, for example, 3 concentrations or 3 replicate each, or a minimum of 6 determinations at 100% of the test concentration. Intermediate precision. Intermediate precision expresses within laboratories variations on different days on different analytes with different equipments, etc. The extent of which intermediate precision needs to be monitored depends on the circumstances under which this procedure will be used. Reproducibility. Reproducibility expresses the precision between the laboratories. The standard deviation, relative standard deviation and the confidence of interval should be reported for each type of precision investigated. Next is linearity. Linearity of analytical procedure is defined as the ability of the given procedure to obtain test results directly proportional to the concentration. This should be evaluated across the range of analytical procedures. Linearity should be plotted as a function of analyte content. It should be established on a minimum of 5 concentration. Initially, it should be established by visual examination of a plot of signals as a function of analyte concentration of the content. If there appears to be a linear relationship, examination of test results should be performed by a statistical method, for example, by calculating the regression line or by the method of least squares. Next is range. Range is considered as the interval between the upper and the lower concentration of analyte in the sample for which the analytical procedure has a suitable level of precision, accuracy and linearity. Following minimum range needs to be considered while validating a procedure. For the assay of a drug substance or a finished product, normally from 80 to 120% of the test concentration. For content uniformity, covering a minimum of 70 to 130% of the test concentration unless a wider range is justified. For dissolution testing, plus minus 20% over a specified range. That is, if the acceptance criteria of a controlled release product is from 20% after 1 hour to 90% after 12 hours. Linearity is to be performed from 0% to 110% of the label claim. For determination of an impurity, from the reporting level of an impurity to 120% of the specification. Next is limit of detection and limit of quantitation, also known as LOD and LOQ. Limit of detection is the lowest amount of the analyte in a sample which can be detected, but not necessarily quantitated as an exact value. The limit of detection is usually expressed as the concentration of an analyte in the sample. In case of instrumental analytical procedure that exhibits background noise, for example like HPLC, ICH describes a common approach to compare measured signals from samples with known low concentration of analyte with those of blank samples. The minimum concentration at which the analyte can reliably be detected is established. Typical acceptable signal to noise ratio is 2:1 or 3:1. The quantitation limit of an individual analytical procedure is the lowest amount of analyte in a sample which can be quantitatively determined with a suitable precision and accuracy. The limit of detection is usually expressed as the concentration of the analyte in the sample. Same approach as described in determining LOD is recommended for LOQ also. Typical acceptable signal noise for LOQ is 10:1. Next is system suitability. If measurements are susceptible to variation in analytical conditions, these should be suitably controlled. One consequence of the evaluation of robustness and ruggedness of analytical procedures should be that a series of system suitability parameters are established to ensure that the validity of analytical procedures is maintained whenever it is used. System suitability testing is an integral part of an analytical procedure. The tests are based on the concept that the equipment, electronics and analytical operations and samples to be analyzed constitute an integral system that can be evaluated as such. System suitability test parameters to be established for a particular procedure depends on the type of procedure being validated. Robustness or ruggedness. It is the ability of the procedure to provide analytical results of acceptable accuracy and precision under a variety of conditions. It shows the reliability of analytical procedure with respect to the deliberate variation in the method. Following are the typical parameters which are varied in HPLC method: pH of the mobile phase composition, different columns, temperature, flow rate, and following are the typical parameters which are varied in GC method: different columns, temperatures, flow rate. Data elements required for validation. Considering the broad variety of validation procedures, it is logical that different procedures are validated using different schemes. Based on the validation requirement, analytical procedures can be categorized as follows. Category 1: analytical procedure for quantitation of major components of bulk drug substances or active ingredients in finished pharmaceutical products. Category 2: analytical procedure for determination of impurity in bulk drug substances or degradation compounds in finished pharmaceutical drug products. Category 3: analytical procedure for determination of performance characteristic, for example dissolution and drug release. Category 4: identification tests. Data elements that are normally required for validation of analytical procedure of each of the above category are as shown on the screen. The validity of an analytical procedure can be verified only by laboratory studies. Therefore, documentation of successful completion of such studies is a basic requirement for determining whether a particular method is suitable for its intended use. Compendial procedures are also subject to regulations that require demonstration of suitability under actual conditions of use. Validation, qualification of utilities. The most important of all utilities are heating, ventilation and air conditioning system that we generally call HVAC system. The HVAC system plays an important role in product protection, personal protection and environmental protection. The validation of an HVAC system is divided into 3 stages of IQ, OQ and PQ. Following are the major tests carried out for operational qualification of HVAC: air balancing and pressure differential test, terminal HEPA filters test, HEPA filter integrity test, pressure differential stress test, control and monitoring devices test, power fail and recovery test, and the performance qualification tests carried out for HVAC are following: temperature humidity control test, air cleanliness test, airborne bioburden test, surface bioburden test. Next is water system. All water treatment systems should be subject to planned maintenance, validation and monitoring. Validation of water system should be consist of at least 3 phases. They are almost same as that of prospective, concurrent and retrospective validation. Phase 1 investigational phase, phase 2 short term control, phase 3 long term control. The water generated using second and third validation phases can be used for routine manufacturing. Alert and action levels. An alert and action level will be usually set to safeguard the water system from exceeding the specified microbial levels. Cleaning validation. Cleaning, like any other critical process, should be validated. Cleaning validation is the documented evidence to ensure that cleaning procedure adopted removes the residues of predetermined acceptance levels. Validation of cleaning method is an important element of qualification and process validation. Cleaning validation ensures that the equipment cleaned as per prescribed procedure will be always within the acceptance criteria set for product residues, detergent and microbial residues. Standard operating procedure should be made available for equipment cleaning. This procedure should be prepared only after completion of cleaning method validation. This procedure should be followed for cleaning the equipment and cleaning log to be maintained. Before the cleaning validation activity is initiated, a cleaning validation protocol needs to be designed. This should take care of all aspects of the study, including the objectives of the study and personnel responsible for the execution. This protocol needs to be reviewed and approved by authorized personnel. Records of cleaning validation, which include all raw data of the test results together with the cleaning record, which should be signed by operator, checked by production and reviewed by quality assurance, should be kept, and a final validation report should be prepared. The final outcome should be started. For example, all the acceptance criteria were met. Personnel or operator who perform a cleaning routinely should be trained and should have effective supervision. The manufacturer should have a strategy on cleaning validation, which should cover product contact surfaces, cleaning after changeover, between batches in campaigns, bracketing products for cleaning validation, periodic evaluation and revalidation of number of batches required should be included. The period and conditions of storage of unclean equipment before cleaning and the time between cleaning and equipment reuse should form part of validation of cleaning procedures. Documented evidence should indicate the routine cleaning and storage of equipment does not allow the microbial proliferation. Equipment should be stored dry and under no circumstances should stagnant water be allowed to remain in equipment after cleaning operations. While discussing cleaning validation, sampling and grouping should be considered. Sampling. There are following methods by which sampling can be done. First is surface sampling, then rinse sampling, and other methods of sampling include batch placebo method, clean in place system and analytical methods. Grouping. Grouping may be allowed under certain conditions. It can be based on grouping by product or grouping by equipment. Grouping by product is allowed for products of similar property, for example solubility, and manufactured on similar type of equipments. The representative product chosen should be the most difficult to clean. Grouping by equipment may be allowed if it is similar equipment or the same equipment in different sizes. An alternative is validating separately by using the smallest and the largest size. Computer system validation. Computer system validation is the documented evidence which provide a high degree of assurance that a computerized system records data accordingly and the data processing complies with the predetermined specifications. The system should be tested, IQ, OQ and PQ should be performed and documented. Results should be reviewed and validation outcome concluded. Summary of validation requirements for computer system are as shown on the screen. So, to get the quality product, each and every step and facility should be validated. Critical factor should be carefully validated in order to validate analytical procedures. Validation is an important part to get the quality products. Thank you.